Okay, it is 3 o'clock, and we have a lot to get through today. So I'm going to go ahead and get started. Welcome, everyone. Thank you for joining us for the spring public webinar for the Gabriella Miller Kids First Pediatric Research Program, also known as Kids First. I know we're all working from home, so please bear with us as we work through the technical challenges that come with running a big webinar like this. Since we have now done several of these webinars in the past, today we will start with just a brief introduction of the program before introducing our featured presenters led by Drs. Mary Marizita and Eleanor Feingold, who will tell us about findings from the Oral Facial Cleft project. Then we will hear from the Kids First Data Resource Center, led by Adam Resnick, who will show us new features of the Kids First Data Resource Portal, introduce you to the cloud-based data analysis workspace, Cavatica, including a presentation from Owen Hershey, who will tell us how he used Cavatica in his Kids First analysis. And then the Data Resource Center will tell us what is on the horizon for further developing the data resource. Finally, NIH staff will provide program and collaboration updates before we address some of your questions. My name is Valerie Cotton, and I am the Kids First Program Manager for the program from the Eunice Kennedy Shriver National Institute of Child Health and Human Development, and I will kick off this webinar with some background about the program. The program was initiated in response to the 2014 Gabriella Miller Kids First Research Act, which transferred $126 million that was in a presidential nominating convention fund to a pediatric research initiative fund and authorized the appropriation of $12.6 million per year for 10 years to the NIH Common Fund, with the first appropriation in 2015. Gabriella Miller, the bill and the program's namesake, was a 10-year-old advocate for pediatric research who lost her battle with a rare and inoperable form of pediatric cancer called diffuse intrinsic pontine glioma, or DIPG, just 6 months before the bill was signed. The vision of the program is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research to uncover the etiology of these diseases and by supporting data sharing within the pediatric research community, which we believe will accelerate research and ultimately lead to improved diagnostics and therapeutics for patients and families suffering from these conditions. The strength of studying these pediatric conditions together is supported by the fact that there are associations between childhood cancers and structural birth defects, as highlighted in publications such as this one from Philip Lupo's group, suggesting there are shared genetic pathways underlying these conditions. However, both sets of conditions tend to be relatively rare and therefore warrant coordinated efforts to build, combine, and study sufficiently powered data sets. Kids First is an NIH Common Fund program overseen by a trans-NIH working group. The Kids First working group is chaired by institute leaders from NICHD, NHLBI, NCI, and NHGRI, with representation from 12 other institutes and centers from across the NIH, as well as the CDC. We have implemented 2 major initiatives that we intend to continue through 2021. This includes identifying childhood cancer and/or structural birth defects cohorts for whole genome sequencing by Kids First sequencing centers, and developing the Kids First data resource to facilitate and empower analyses of these data sets. We are not yet ready to announce new initiatives for the last 3 years of the program, but today we will tell you a little bit about our progress on these initiatives, related efforts, and additional goals on the horizon. We've had 5 cycles of cohort sequencing, which represents nearly 40 projects, 15,000 patients, and 37,000 genomes, currently being sequenced by the Broad Institute and the Hudson Alpha Institute for Biotechnology in collaboration with St. Jude. 10 Kids First data sets are currently accessible through the data resource, and this number is growing. The Kids First investigators who contribute cohorts for sequencing and data sharing, as well as the sequencing centers and the Data Resource Center, form the foundation of the Kids First research community. But this community is growing too, as these groups form new collaborations and as external researchers access and analyze Kids First data. Anyone can register with the Kids First Data Resource Portal to search across Kids First data sets. Since launch, more than 1,500 users have registered, and more than 100 dbGaP data access requests have been approved for analyses of genomic data. And today, you are going to learn about how the components of the Kids First data resource can foster these community collaborations to further accelerate research and discovery. And with that, allow me to introduce our first presenters today, Drs. Mary Marizita and Eleanor Feingold from the University of Pittsburgh, Dr. Marizito's research is focused on human genetics of complex traits, while Dr. Feingold studies statistical and computational methods of genetics. You will also hear from Dr. Elizabeth Leslie, currently at Emory University, whose research is focused on the human genetics of orofacial cleft disorders. And finally, Dr. Harrison Brand from the Broad Institute, who studies the application of computational genomics in congenital anomalies. This team has been with us from the beginning as one of the earliest recipients of the Kids First Exon 1 cohort sequencing program to study the genetic component of orofacial clefts, and today we look forward to hearing about their progress. So with that, Mary, I will pass the presenter role to you.
Okay. Hi, Valerie. I'll just share my PowerPoint. Can everybody see this?
Yes, I can see that. Okay,
great. So thanks, Valerie, and the whole DRC and Kids First staff for inviting us to present at this webinar. We have been with Kids First from the beginning, and we're very proud of that. So we're excited now to show you some of the progress that we're making. The goal of our research collaboration is to understand the genetic basis of orofacial clefts, which is one of the most common structural birth defects in humans worldwide, and it's the most common craniofacial birth defect. It appears in all populations and has a variety of phenotypic expression. It happens early in fetal development, starting at about week 4 through weeks 10 to 12, that the lip and palate develop. So it happens, in other words, often before someone knows that they're pregnant. And they occur in 3 major forms: cleft lip alone, cleft lip with a cleft palate, so the lip and the palate are involved, and cleft palate. And they are traditionally grouped into 2 major categories: cleft lip with or without cleft palate, and cleft palate alone. So our collaborations are worldwide in OFC and have been proceeding for many years with a number of important underlying principles. So there's differences by race, population, and ethnicity in the epidemiology of these birth defects. So it's important for our projects to have a multi-ethnic and global focus. We are particularly interested in deep phenotyping and, of course, understanding genetics. So we have a deep phenotyping protocol where we ascertain certain data by questionnaires such as demographics, personal and family medical history, pregnancy history. We do a detailed physical examination to really understand the individual birth defects, oral exam to look at the dentition, speech sample, etc. And then we derive a number of types of imaging like 3D facial images, intraoral photographs, ultrasounds, etc. that allow us to derive other types of traits as well. So as far as GMKF is concerned, we have had 5 projects approved so far, which total about 1,400 OFC trios to date. The first one that we had funded was a project of trios of European descent, then a Latin American, specifically Colombian project, then a project from Africa and Asia that was led by Aziz Boutali and Terry Beatty, and then an Asian study, specifically Filipinos, that was approved and samples were starting to be assembled, and then we've been delayed with COVID-19. So hopefully as soon as we're back, we'll be able to send those samples in. We also have 1 additional project that's under review for additional Latin American trios. Now that we have all this incredible sequencing data, we have a large collaborative group that's working on analyzing the projects. Our first paper was published at the end of 2019 with under the leadership of Nandita, and it reinforced a region that had had some suggestive results in the past, but is a novel region that's associated in the Colombian population. In the African and Asian analyses, they are also kicking off with a variety of under a variety of approaches. And one of the interesting recent results is that a particular signal that was felt to be seen only in Europeans actually has some significant evidence, a region at the end of chromosome 8. So it's coming up as positive in the African population. Then we have a lot of ongoing analyses, and I really appreciate Dr. Leslie and Dr. Brand joining us to walk us through what they've been working on. So I literally could have given this whole introduction just thanking everybody. So here's just a quick shot, and I will turn it over to Dr. Leslie. Or Valerie, will you turn the presenting over to them?
Yes, I'm handing it over to Dr. Leslie right now.
Great.
All right. Valerie, can you see that?
Yep.
All right.
Sounds good.
So I am going to share an update on our ongoing efforts to understand the role of de novo mutations in orificial cleft risk. And as a brief reminder, de novo mutations are a class of variation that occurs only in the child and is not found in either parent. Everybody has these variants. There are about 100 of them in a single genome, and 1 out of the 100 is going to hit a protein coding gene and be part of the exome. And these coding de novo mutations are known to be common causes of both congenital and developmental anomalies. And there's pretty good evidence in autism, congenital heart disease, diaphragmatic hernia, and others that de novo mutations are enriched in these cases. And one of the things that is similar between these disorders and clefting is that clefting can occur sporadically. Only about 15% of cases have a positive family history. And so that suggests that de novo mutations could play a role. But there haven't been genome-wide studies of de novo mutations, and the only evidence that had existed previously were a handful of smaller sequencing studies through candidate gene or small exomes to support de novo mutations. And so our goal has been to use this Kids First data to understand genome-wide what the contribution of de novo mutations are in orificial clefting. And so this has been the work of Madison Bishop, who is a postdoc in my lab. And for today's talk, I'm going to share the results from her 3 main questions. First, are coding de novo mutations enriched in OFCs? And what is the biological relevance of those de novo mutations? And finally, is there a clinical significance or relevance of these variants? So today we're just sharing the results of 3 of the populations that Mary talked about. We analyzed a total of 756 trios after QC from the US, Colombian, and Taiwanese Kids First cohorts. Most of these trios have cleft lip and palate. There are about 58 trios that have cleft palate only, and that's going to come into play very briefly. Overall, we identified just over 73,000 de novo mutations, and today, as I said, we're going to focus on the 862 of these variants that occur in the coding portions of the genome. So the first thing Madison did was annotate these variants, and about 12% of them are what we consider predicted loss of function variants. They're stop gain, nonsense, or splice variants. About 70% are missense variants, and we grouped into this in-frame insertions and deletions. And throughout this presentation, we're going to use this blue and red color coding to refer to these groups of variants, and then combine them into a group called protein-altering variants that will be represented in purple. And as a point of comparison, we're going to use the 24-25% of variants that were synonymous, because this is the set of variants that are not likely to be contributing to risk of clefting as a group. So overall, we see an excess of protein-altering de novo mutations in clefting. We do not see an excess of de novo mutations among synonymous variants. And you can see that the effect size, there's a bigger excess of de novo mutations that are predicted loss of function than missense. and the excess is overall something about 1.2-fold. If we split by cleft type, you see a very similar picture for cleft lip with or without cleft palate. That's the bulk of the data. And what is interesting is that even though we only have 58 cleft palate trios, we still see evidence of an excess, although our confidence intervals are quite large. But this is encouraging that that group of clefts also might have a role of de novo mutations. And then finally, we split by sex. So we know that there is a 2-to-1 prevalence ratio of males to females. So males are twice as likely to have cleft lip with or without cleft palate. And we thought that perhaps de novo mutations might contribute to this difference. And at least in this data set, we don't see any evidence for that. So there doesn't seem to be a difference in the distribution of de novo mutations by proband sex. If we do gene set enrichment analysis, we see that de novo mutations are enriched in developmental genes. What we highlight are a number of morphological processes, human cleft palate genes, but overall, we sort of hit on sort of broad categories of morphogenesis and developmental problems. And so we wanted to get a little bit more specific. And so we turned to a recently published craniofacial specific data set where the developing mouse lip was dissected from the embryo and then single cell sequenced. And through 2 rounds of clustering, the authors identified clusters of ectodermal genes and mesenchymal genes. And we hypothesized that de novo mutations in clefting might be enriched in the marker genes of these cell clusters. And so what we can see here in this sort of radar plot is that we have a significant excess of de novo mutations in 2 of these ectodermal clusters. And again, as in the overall picture, there's a stronger enrichment among loss of function de novo mutations. And as it turns out, these clusters are at the point of fusion of the upper lip. So one is in the olfactory epithelium, and one is in the cells that are at the very point of fusion or undergoing fusion themselves. So that was very exciting for us and really emphasized the biological relevance of these variants. We also did an analysis to see if there were any individual genes with and on the top are the genes that have predicted loss of function de novo mutations, and 2 genes reached exome-wide significance, CFAP2 alpha and ZFHX4. And then if we consider all protein altering de novo mutations, again, there are 2 genes that reach formal significance, IRF6 and TFAP2 alpha. So that was also very exciting. It was also a little bit disappointing. And so we wanted to use a data set that would and a way of analyzing the data that would let us get at all of these genes that have de novo mutations, but maybe only have 1 de novo mutation and don't reach a formal level of significance by our statistical tests. So we used a bulk RNA-seq data set from cranial neural crest cell lines. These were reprogrammed from human iPS cells by Joanna Wysocka's lab at Stanford. And what we did was we ranked all of the genes based on their expression level in these cells, and then ranked all of the genes on the y-axis based on their constraint scores for PLI. And the overall hypothesis then is that the upper quadrant of genes, those that are highly expressed in cranial neural crest cells and are constrained to loss of function variants in data sets like NOMAD, would be enriched for mutations that might be causal for orofacial clefting. And so if we zoom into that quadrant, that is exactly what we see. There is about a 2.5-fold enrichment of de novo mutations that are predicted loss of function in this quadrant. There's no excess of missense variants, as it turns out. And some of these genes, again, are ones that we saw as individually significant. AP2 alpha and ZFHX4 are among this group. But what was more interesting is, so we took that group of genes and again ran it through a gene set enrichment analysis. And what we found is that among the 29 genes that had predicted loss of function de novo mutations that were in this upper quadrant, 8 of them were predicted to interact with SOX2. And that was significant. You can sort of expand that a little bit by looking at any of the genes with protein altering de novo mutations. And there were 16 genes in this group that were predicted to interact with SOX2. And these are just predicted interactions. They haven't been experimentally confirmed and come from a variety of different data sets. But it's intriguing, at least, that a set of genes like this would come out as significant, even if individually we don't see an individual excess of de novo mutations in each of these genes. But again, ZFHX4, AP2 alpha are in this group of genes, and so that was also exciting. And then Madison's other question was whether there's a clinical significance for clefting. And currently there are a number of companies that do clinical sequencing and clefting. The size of their panels is very variable. It ranges from 6 genes to 172 genes. And so we wanted to sort of make our own panel and then assess the evidence. So we assembled a list of about 300 genes that are on these panels or identified by GWAS or through OMIM to see what the excess was. And what we see is that, as before, there is an excess of de novo mutations in these clinically relevant genes. In particular, we had an 18-fold excess of predicted loss of function variants in the list of genes that where mutations cause autosomal dominant syndromes. There's also an excess of missense variants in that set too. And so this tells us that there is a clinical relevance here, and maybe a reason to do clinical sequencing and clefting. And again, some of these genes are very familiar to us in the clefting community. AP2 alpha is one, IRF6 is another. We expected to find multiple mutations in some of these genes, but we weren't expecting to find this number of variants. And this has big implications for the genetic counseling of families that have kids with clefts, because they may not have been counseled in a manner that would be consistent with maybe autosomal dominant inheritance pattern for future generations. So in summary, we identified an excess of protein altering de novo mutations in our cleft trios, about 1.2-fold more than expected. So this was a modest excess. We know that these de novo mutations are in biologically relevant genes. These are present in marker genes for cell clusters that are at the point of fusion of the developing mouse lip. And they are among the genes that are in the top 20% of human cranial neural crest cell expression that are also then constrained to mutation. We found de novo mutations in several clinically relevant genes. Overall, it was about 18 times more than we expected among autosomal dominant clefting genes. And 3 genes in particular had individual excesses of de novo mutations, IRF6, AP2 alpha, and ZFHX4. IRF6 and AP2 alpha are sort of known genes to us. The number of variants we found in AP2 alpha was surprising. But IRF6 was sort of expected. But ZFHX4 was a completely novel gene for us. We identified 2 loss of function de novo mutations, and we know that that gene is expressed very strongly in the developing face. And so we consider this evidence that ZFHX4 is a novel gene for clefting based on this study. And so overall, coding de novo mutations do seem to play a role in clefting, although they are not a sort of the major cause. The enrichment is not as strong as it is in other birth defects, but nonetheless, it does play a role. And this is work that was done mostly by Madison Bishop, who I said was a postdoc, but also Kim Diaz-Perez, a graduate student, and Samantha Ho, an undergraduate student who helped with the gene panel assembly, and a number of other collaborators. And finally, I'll say that the manuscript reporting these results is on bioRxiv now. And so for more information or other details that I didn't share here in this limited amount of time, you can find in our bioRxiv preprint.
Great, thank you, Dr. Leslie. I'll hand it over to Dr. Brand. Harrison, I don't see your slides yet. Oh, here they come.
Okay, okay, got
There
it.
you go.
All right, we good?
Yep.
All right, perfect. All right, hi everyone. I'm Harrison Brand. I'm a new faculty member at MGH, Harvard, and the Broad, and I'm really excited to present my work today on calling structural variation in this oral facial clefting cohort. So the impact of structural variation in non-syndromic forms of orophacial clefting is really uncharacterized and hasn't been explored too much. And so I was really excited to look at that with this cohort. So for this study, we applied GETK SV, which is a computational SV discovery pipeline. We applied this on over 2,500 of the samples from this cohort and 837 complete trios. I was able to call structural variants from 3 different GMKF studies and all 4 of the available OFC populations. And one of the advantages that we have using GASV, which is a method which is our structural variation calling method I'll get into later, is that this was recently applied in the NOMAD AD reference database. And so therefore we have a reference cohort that actually has these same exact populations that we can control, that we can compare structural variations against, which we can use for filtering later when we're looking for disease relevant variants. So I know that there's a diversity of backgrounds in this audience, and so I wanted to just do a really brief background into what structural variation is. So structural variants are alterations of the genomes that are greater than 50 base pairs. You have deletions and duplications, which are collectively referred to as copy number variants. So those are losses and gains of the genome. You also have inversions, which are flips in the orientation of the genome. And then you have insertions where a piece of the genome is inserted somewhere else. Okay. Now, in addition to the standard structural variants, you also have combinations of combinations of these 4 types of basic SV classes to form complex events, such as this inversion with flank duplications, as well as this inversion with flank deletions. So it's important when we're looking at structural variants to consider all types of variation, not just the standard types. Now, as we've expanded SV calling into these large reference cohorts, we found that complex SV are actually rather common in the genome. And in this example, we have calls from the NOMAD cohort. And in less than 15,000 samples, we're able to resolve over 5,000 of these complex structural variants. Here's an example of many of the different kinds that we find. And I just want to highlight that, given the frequency of these are really important to consider in whole genome sequencing research studies. So I'm just going to give a quick background on the methods that we use. So this is the type of information that we can use to call SV from whole genome sequencing. Different SV require different evidence types. Now, what's important about this is that no existing SV caller uses all forms of information, all forms of this information to detect SV. And so there are many different SV callers, and each has their own strength and weakness. And so to overcome this fact that none of them actually use all this available information, we developed an ensemble approach that takes advantage of multiple callers to really maximize our discovery power and take advantage of the strengths of each individual caller. And so what this pipeline does is it runs several unfiltered algorithms, existing algorithms, to maximize sensitivity. Now we've selected this set we've selected a set that really complement each other. So these are the 5 algorithms we run, but it's very flexible and it can run other algorithms as you wish. So after we run these algorithms, we then reevaluate the SV calls from each of them using information that's directly assessed from the BAM. And this is meant to reduce positives and to improve specificity. After we've which variants are real, we then resolve them. And so by doing this, we can capture both unbalanced events like copy number variants, balanced events like conversions and translocations, as well as complex structural variations by integrating these different signature types. Importantly, this method has been adapted to work on the cloud. And so this can be assessed on the Google Cloud via the Broad Institute's Terra platform. So one of the first things that we like to investigate when we're looking at structural variants in a cohort is the sex chromosome aneuploidies, as well as a gender check, just to make sure that the dosage of an individual lines up with what we expect based off of a given file. In this case, we can see that we have 5 sex chromosome aneuploidies, which given this cohort size is right in line with expectation. 2 of these are parents and 3 of them are fathers. So after running GTK SV on this cohort, we found several 100,000 total variants. And this equated to about 8,000 to 9,000 SVs per person. You can see that deletions are the most common, followed by duplications. This is right in line with our expectations. The number of S sites and samples can change based off of filtering, and different ethnicities have different amounts of SV. But this is right within our range that we usually see, which is between 6,000 and 10,000 variants per cohort, or per person in a given cohort. So a cohort such as this, and Elizabeth really highlighted this before, we want to start investigating, we typically start investigating de novo events, which typically have the highest yield of pathogenic variants. So similar to when you're working with SNVs and indels with a tool like GTK, we still have to do careful filtering for our variants, just to ensure that we have high precision of de novo variants, because we don't want false positives that are going to muddy our analysis. Some of the reasons why we may have false positives in our RC pipeline is due to things like parental mosaicism or just lack of phasing. So it's very difficult to find certain compound heterozygous events in these individuals. And so a parent may have a overlapping deletion and duplication and look normal to us, but then when we look at the child, we see one of those events and it looks like it's not in the parent, but then when we actually go back, we can see that in fact the parent actually had a overlapping deletion and duplication, which looks has a normal copy state. So in order to kind of get around this these potential false positives, we developed GTK de novo workflow. And another important thing about calling de novo SV is that the number and the amount you get is really based on your sample phenotype and ascertainment. So here's the de novo pipeline that we developed. The first thing I want to highlight is that we work from an unfiltered VCF. Just to make sure that we maximize sensitivity, so we're sure that we're not missing any potential de novo variants. And so when we start, we typically have around 4,000 de novo SV per person. After removing unresolved events and CNVs that have multiple copy states, as well as SVs that have a frequency of greater than 0.5% in parents, we're able to dramatically reduce that number down to about 100 de novo SV per person. Given that we expect less than 1 per person, we know this is still way too high, and so we have to keep filtering these events. So we came up with a set of stringent filtering criteria that can be applied all computationally. And when we do this, we get a high confidence de novo set in 8% of samples with at least one de novo SV. But we know based off of previous studies that that's a little bit low. And so we next apply a less stringent filter, and we manually check some medium quality variants. And so in this case, we manually investigated a little over 1,000 variants by hand. And by doing this, we were actually able to double the total number of de novo SV we found. And so in this call set, we had a final amount of 165 de novo SV, and about 1 in 5 individuals had at least one. So here's the composition of our de novo SV. So the majority are copy number variants, and well, about a quarter have a loss of function effect, but still the most common predictive functional genic impact is to have no effect at all. Next, I just want to walk through some highlighted de novo SV. So one of the first thing we look at was whether any of our de novo SV hit a site that is known to be susceptible to novo mutation. So here's a list of reoccurrent sites, reoccurrent de novo CNV that we found in this cohort. So we have had 14 of these. Now, most of these are these are all known syndromic or microdeletion sites, and most of these are not necessarily affiliated with cleft, but they do have cases that are reported to have cleft. In a few of these, we did not find any cases in the literature that have cleft, so these are certainly interesting variants to follow up on. Moving beyond these known reoccurrence sites, we also find other large CNV, so they're greater than meg. An example of a 5.3 meg deletion that disrupts greater than 30 protein coding genes. Now, when you're looking at this event is likely causative and related to the orofacial clefting, but it's really difficult to pinpoint, which individual gene is causing this or which group of genes is causing this. So when we focus on smaller gene disrupting SV, we actually do find some that are known to disrupt established orofacial clefting genes. So here's an example of a deletion that disrupts CTND1, as well as one that you'll recognize from Elizabeth's talk that disrupts TFAP2A. Now, what's interesting about these is that these both have already been reported to be associated with orofacial clefting, and these are also both the only genes that were disrupted by SV that were also disrupted by a loss of function mutation in the SNV and endo call set from that Elizabeth generated. So looking beyond established OFC genes, we have several CNV that disrupt genes that are known to have support in animals, but not necessarily have evidence of human support until this point. So here's two examples of that. Now, back before affordable sequencing and even really cheap array-based SNP genotyping, research groups would pick genes of interest to do candidate polymorphism studies. And in this cohort, we have 2 de novo SV that disrupt such genes. And in this case, we have a intragenic exonic duplication of RFC1. Now, this gene is, this CNV is predicted to copy a few exons of this gene. And so therefore, it's likely, we predict it to be protein disrupting. And it should be noted that this gene is involved in the folate pathway, which it makes a ton of sense to be related to OFC. And moving beyond constrained gene, or moving beyond established OFC genes, we do have a few interesting novel candidate genes. Now, we have 7 genes, we have 7 CNVs in this cohort that disrupt constrained genes defined as NOMAD LUF score less than 0.4. Here's an example of a 8KB deletion that disrupts NRF1. Mutation in NRF1 are not yet associated with OFC, but it does make sense. The gene makes sense that it could be involved, and it's a transcription factor of small pathways that could contribute to OFC. I also like to highlight a complex event we found. While this is not necessarily pathogenic, it is interesting in that it is an insertion event where you have two inserted pieces that are inserted from downstream over 60 megs away. And these are fairly large events, 174 kb for one, 464 kb for the other. And you'll notice that there are actually 2 meg apart within them. So you have 2 separate inserted pieces, as well as a deletion at the insertion site. Now, in addition to these complex sites, we're also able to find reciprocal translocations. And this is completely from the genome sequencing. So we do not have a karyotype, which is the classic way of finding these. In this case, we have a disruption of this gene CNIHR3 from this reciprocal translocation. This is interesting because it overlaps a 1q41, q42 deletion syndrome, which is cleft is a known phenotype, as well as a few cases have shown cleft lip. And so this could be pointing to a gene in that microdeletion syndrome that is involved in the orofacial clefting. And then I'm running a long time, so I'm just going to really quickly go over this. But we also have the ability to detect mosaic events. And here's 2 examples of a mosaic deletion and a mosaic duplication. So with that, I'll wrap up. We have application of GTK-SV was able to discover a diverse set of SV. We were able to use de novo filtering to adjudicate SVs down to about 1 in 5 individuals. We find both established OFC genes and novel candidate genes for the follow-up. And whole genome sequencing has a resolution to detect both complex SV and balanced SVs, not easily detectable by exome sequencing or microarrays. So in the future, we'd like to explore rare inherited SV, do some non-coding exam. We need to examine non-coding SV. We are working on integrating results with SV and indels. Also need to investigate recessive compound heterozygous variation. And we're also very interested in applying GTK-SV to additional GMKF cohorts to build an aggregated SV map of congenital birth defects. So I don't have a lot of time, but this was a group effort, and I'd like to thank the Broad GMKF and SV sequencing analysis teams, as well as the GMKF OFC working group, which was great providing feedback. So thank you.
Thank you so much, Harrison. I think there's actually a question for you in the Q&A box. So feel free to type a response to that question, but for the sake of time, we're going to move forward. Now I will hand it over to the Data Resource Center, led by Adam Resnick from the Children's Hospital of Philadelphia, in collaboration with multiple other sites. Dr. Resnick is the co-director of Data-Driven Discovery and Biomedicine at CHOP, and his team, including Allison Heath, also from CHOP, and Vincent Ferretti from St. Justine University Hospital, is going to show us some developments with the Data Resource Center.
Thanks so much, Valerie. So, really, that was a tour de force, set of presentation that, we just saw from Dr. Marizita, Leslie, and Brand. And, one of the things that we've been thinking about within the Data Resource Center really has focused on how do we empower scaling these types of discoveries. You saw, for example, in Dr. Leslie's work, the need to really integrate other data sets and other resources with the Kids First data sets, being studied. Likewise, in Dr. Brand's presentation, you saw really the need to really develop ways to prioritize and manage the large-scale data that comes with whole genome sequencing and the application of filters. And so within the Kids First as a Data Resource Center, if you just review sort of our current state by the numbers, and Valerie highlighted this to some extent, the scale really dramatically becomes apparent. As Valerie mentioned, we have about 10 studies at this point in time. This represents more than 10,000 participants from 3,000 families across 7,500 diagnoses with more than 70,000 different phenotypes across more than almost 11,000 genomes with more than 1 petabyte of data. And the key here is really to leverage these data sets and resources in ways that are very similar to Dr. Brand's sort of analysis of really, highlighting how to extract and maximally utilize the data on behalf of the discovery process, both within a particular syndrome or disease context, but especially for Kids First, the programmatic emphasis on leveraging cross-disease analysis and then spanning across pediatric cancer and structural birth defect as a discovery tool. So we're excited today, and I'll turn this over to Vincent to really release a couple of different additional portal features that have really been part of our strategic development within the DRC on behalf of really empowering the kinds of analyses and discoveries that you just heard about by leveraging portal features and platform features for the Kids First community. So it's all yours, Vincent, as Valerie hands it over to you.
Adam. Can you see my screen now?
Not yet. I think you'll have to hit the share screen button. Yes, now I can see it.
Okay, so let me go in presentation mode. So I want to get rid of this thing. Yeah, so today I would like to talk to you about major feature development in the portal. The first one is about ontology. So we have developed a new ontology browser that I'm going to show you in a live demo in a minute. And then the second feature, but not the least, is a new germline variant data warehouse that we have been working on for the last 2 months now. And then I will conclude with some discussion about next step and future direction. So the ontologies within Kids First. So Kids First, from the beginning, Kids First has been making extensive use of ontologies as a means of harmonizing clinical data across cohorts. So currently, Kids First is using the human phenotype ontology for phenotypes, HPO, Mondo for diagnosis, NCIT, and SNOMED as well. So ontologies provide 2 things. First, controlled vocabularies, and secondly, relationships, parent-child relationships between the terms of an ontology. For example, in the HPO, you have this kind of relationship. Oral cleft is an abnormal oral cavity morphology. This is a relation. It doesn't have to be the verb to be. You can have other ontology with as a car, as a wheels, as wheels, and so forth. So but in HPO, it's always the same kind of relationship between terms of the ontology. So until recently, we didn't fully explore this relationship for participant searches. So the user could select specific terms from a flat list of possible value that is shown here on your right, and then search for and then find the participant tag with this exact corresponding terms. But the problem is that cohorts annotate their participant using different levels of precision. So therefore, it's really important to be able to find participant with precise annotation across cohorts using broader search criteria. And this is what we have been working on. So we modified the data model to integrate ontological relationship as participant search criteria. So for example, now user can now find participant with a specific term and all its descendant as well. So if I go back to my example above, so if you search for participant with abnormal oral cavity morphology, you will find as well oral cleft participant, because there is this kind of relationship between the 2 terms. So I'm going to show you a very quick demo of this feature. So let me escape. And then I go to the portal. So this is our cohort builder. So it's used to find participants of interest in KidsFirst. So at the top, you have a menu of search criteria, for example, ethnicity, that you can and combine into one or more queries in this section in the middle. And then at the bottom, you have the results, which is a participant list. But we provide two views for this result. So you have a summary view, which provides some aggregation and breakdown. And you have the table view, which provides the explicit list of participants that meet the criteria of your query. So let me do some queries to illustrate the new phenotype browser that we have built into this code builder. So let's say I'm interested to build a query involving some phenotypes, HPO phenotypes. So I can find usually I use this text box, search text box on the left, and then you can see the observed phenotype here. Click, and then I get a flat list. The flat list I mentioned 2 minutes ago of phenotypes that are present in my entire KidsFirst cohort. So you can see you do have a lot of phenotypes here. And then you can select some of them to build a query. But another way, if you really know what you want to search for, you can actually type directly the value of your phenotypes here. So let's say I'm interested in art valve morphology. So abnormal art valve morphology. You can see here that there are 460 participants with such a phenotype. So I can select them. And then indeed I get 460 participants. If I look back to the summary view, there's an issue here. I don't know why. Let me start again. Okay, so let's do it again. I'm sorry, there was an issue here. So that's art valve. I'm sorry. Okay, I see. Sorry, I was looking at the wrong. So okay, sorry. Let's go back. So I added this term, and then you can see that only one study actually as participant. The 460 participants belongs to a single study, which is the KidsFirst congenital heart defect. And if you want to verify that, you can start a new query if you want, and then you can select all the studies but this one. So you can select all, and then you see the second one is actually the congenital heart defect study. I remove this selection, apply. And I have 10,000 participants, 10,500 participants with for all the studies but heart defects one. And then I can do a combination, a hand. And as expected, I have found no participant that have both this phenotype and that are belonging to the KidsFirst to all the studies except the KidsFirst congenital birth defects study. So let me duplicate this query, and so it's not a surprise actually that let's say this study, the congenital heart defect, annotates a subject cardiovascular condition or phenotype with high precision. But we wouldn't expect the same level of precision from other birth defects such as orofacial cleft or from a study from cancer for cardiovascular phenotype. So, but it doesn't mean actually that other cohorts don't have patients with abnormal heart valve morphology. And this is why you might find them because you want to maybe explore the genomic commonalities between these patients and the abnormal and the congenital heart defect patient. So this is where the phenotype builder, phenotype browser comes into play. And to access this, you can go here and you open here, and then you see on your left side all the HPO ontology actually, and the terms that are represented by at least one patient in the kids first cohort. And on the right side, you have the one that have been selected, so the abnormal heartbeat. And here in this list, on the right side, this column is the number of patient or participant who have the corresponding term. So you can open the tree, you can see the counts decreasing as you go further down in the hierarchy. And then that's it. Okay, now let's say I want to find this heart valve morphology. Find them here. And then you can see that this term is actually a child of the, it's actually a cardiovascular abnormality of the cardiovascular system. So let's select this and add this term on the right side. And then you will see that I have many more patient participants. I have 676 participants with abnormality of the cardiovascular system. Compared to 460 for a more specific term. And then I can see that those 600 participants are part of 4 studies, the initial one, the heart defect, but also 3 other studies. So let's add this one. So I can find 41 participants with abnormality of the cardiovascular system within the study. So this way, and now if I go back to, I can reopen the phenotype browser for this 41. And now you see that the trees are a bit smaller actually. And then this again represents all the phenotypes in this 41 participants. So I can, if I want more participants, I can actually add more terms. Let's say I add the abnormality of the musculature. After all, the heart is a muscle, so you can click, add it. And then you get 133 patient participant with this, this characteristic. So this is the way we will use the phenotype browser. It's really to find phenotypes across studies, kids first studies. Now, the next step for this would be to analyze the variants that we have in this patient. And this is the second topic of my presentation today. It's a new variant database that we have created. But before going there, let me show you something. So if you go to the table view, actually the next step, if you want to analyze this set of patients or participants, you need first to analyze and find the variants, do some query with the new variant database. You need first to save this set of participants in your workspace. So you can save participant, you can name demo set, for example, and then you can save. I won't click save in it because I already saved it, but this is the way you actually move data from the cohort builder to our new variant database for further analysis. So let me go back to the presentation very quickly. I want to talk at you about the variant data within Kids First. So currently the variant data is available in file format, in VCF files. And these files can be searched using our portal file repository. And then once you have found the files, the variant files of interest, you can push them to our compute cloud infrastructure, which is Cavatica, and then start doing some analysis on these data sets. So you need for that, of course, to be skilled bioinformaticians. So it's not given to everybody. And this is why we decided to develop a data warehouse for these variants so that we can access these data sets directly from the portal and without knowing, without necessarily being really high-skilled bioinformaticians. Okay, so that's the goal of this project. So, so we have built a performant and scalable variant database, again, that can be queried directly from the portal. This database will include a comprehensive set of variant annotations, so genes, allele frequencies, gene panels, inheritance, and so forth. We will integrate the clinical data to this database, so you will be able to do multi-dimensional queries involving both clinical and genomic criteria. I give an example here: find all rare misvariant with high functional impact in low-grade glioma patients affected by any cardiovascular abnormality. So you see, I mix cancer, birth defect, genomic properties of variants, and annotation. And on the top of this database, we will build web-based variant analytic tools and visualization tool that will be integrated into the portal. And of course, all this thing is at its core, it's secure and will enforce security and privacy rules. So that user can only access variant data set they have been authorized to, because germline data is a kind of a control tier data set. So this is a quite big data challenge because we do have a lot of germline variant in kids first. In our first version, we have 8 studies, almost 8,000 participants, more than 8,000 participants, and more than 250 million unique variants, and 42 billion occurrences. So a variant within a participant. We have 42 billion occurrences of variant in participants. So we have internally another version with 11 studies, and we reached already 70 billion of occurrences. So just by adding 3 studies. For comparison, you can see that, for example, the NCI, the Generic Data Common, GDC, has 3.1 million somatic variants only for 10,000 cases. And the ICGC, International Cancer Genome Conference, has 82 million somatic variants for 19,000, almost 20,000 cases. So one other complexity is not only the size of the data, but it's actually the complexity of the data, because we really want to build responsive web interfaces and that allow to again elaborate this kind of multi-dimensional queries involving clinical and genomic data criteria. So this is quite complex, and we're not there yet, but we're working hard on this. So very quickly again, so this is our workflow. So on the left side, you have the data sources. So for the first one is, of course, the files that are stored on AWS, the Amazon. These are the GVCF that I were talking about a few minutes ago. We have all the participant clinical data that can be browsed into from the code builder in the web portal. And we import as well public annotation variant like TabMed, ClinVar, and so on. All these 3 data set data sources are loaded into what we Spark cluster. I won't go into detail here, but Spark, it's a kind of scalable technology, so you can add more nodes as your data grow, and then you will obtain the same kind of performance. So we import this in Spark cluster. We do a lot of transformation. We load this data into a Parquet files, which is another big data technology to use for this kind of project, actually. And we also, on our cluster, we install or we deploy a Zeppelin notebook. I'm going to talk to you about this in a moment. And this blue box actually it will be accessible from directly from the web portal for actually performing queries on the variant database. And this is the thing I will demo you in a few minutes. And once we have this, the next step will be to index using another technology called Elasticsearch, which is very, very fast and is meant to be run behind the scene of any web interface, basically, because as again the web interfaces requires a lot of space. You want responsiveness, so this would be ensured by indexing this parse cluster using Elasticsearch. So the first, today we're talking about phase 1, and we're going to release it in a few weeks. And the phase 1 objectives is really to build the foundational infrastructure of this variant database, to import a first set of studies, and to annotate the variants. So we had to create this annotation pipelines. To implement the data security framework right from the start, and to build this deploy this kind of workspace environment, Zeppelin, for user to work on the data. And yeah, so let me do a quick demo. So this is the new menu item that we have on the menu bar, variant DB. You click on this, and then you get the kind of very simple home page for our new journal. You have a summary, some numbers here that summarize the content of the first release. We have 8 study again, and almost 251 million distinct variants. And then as a user, you can launch your own Spark cluster. So when I click on this, a lot of things happen behind the scene. It's quite complex, and I want to thank actually Jeremy Constanza and Alex Luxcancy that I work hard to make this happen. It's basically when you click on that, the user will launch his own private individual Spark cluster, and then which will have access to the variant database. So let's do it. And this is what you get when you launch your own cluster. So these are what we call notebooks. So what the notebook is, it's very simple. It's a file, a text file that contains pieces of code that can be executed individually directly from the web browser. Okay, so you have files, and these files are organized in directories as usual, and so forth. So I prepared one file, one notebook for this demo called webinar. And this is loading now. So I won't go too much into details, but just to show you the principles and some examples of queries or analysis that we can do with a notebook. Okay, so you see those sections in Zeppelin. It's called a paragraph. You can add a paragraph, and this is where you type your code. And nicely, Zeppelin support a lot of programming language. You can write your code in Python, SQL, R, Scala, and many others. So you're not limited to one language to access the database, the variant database. Also, within the same notebook, you can have different paragraphs with different programming language, and which is also very convenient because some programming language are more convenient for some tasks than others. And also you have the markdown language, which is just used to write text, like to create comments and add section into your notebook. So this is what I did here. So my first title section is clinical data, and this is just to show you that actually the variant database also includes on our participant in kits, so that you can join this data with the genomic variant database. So this is the second here, for example, is number of participant per studies, chart like this. And this is a SQL query, so you don't need to understand that. You can hide it, actually. I will. And then you have this. And you can create pie chart, but you can also create bar chart for the same. You can have the table. You can download the results, and so forth. So a lot of possibilities that you can do within a paragraph once you have the code written and executed. So here it's the way you execute your code. So you execute this query again, it's running, it's asking, and this is real time. So there's a record, and then you get the result. Okay, so that's the principle. Now I go down. So this paragraph is to show you actually the list of available save set that I saved previously in my code builder. You can see that they're all available here in my variant database, and you can see the demo set here as 120 23 participants. This is the set, the set that I created with you a few minutes ago. So I do have a lot of set. And now, if you want to join this with phenotypes, for example, you can even create simple form to input parameter in your code. So you see, I can add the demo set, save set. So this gives you the list of the number of participants in a given set for observed phenotypes. So you have the here, for example, this is the weakness of Pachymer, broken down by male, female. Okay. You can, let's say I use another set, key F cardiac. Can run the query again, and then I obtain another breakdown. Here you go. Again, you can do you can export this in table and so on. You have a different view of a notebook. I would switch to the simple view, which merge all the paragraphs together, so it's cleaner. So the second sensor is about the variant data. So this again to illustrate you the kind of queries we can do with this database. So this paragraph is about the number of variants per gene consequences. So again, we have 251 million. If you can, I can run this query again, and then I see that the vast majority of them, 120 million, are intron variant, intergenic variant, non-coding transcript variant. And if you go to the end here on the right hand side, you have the missense variant. Missense variant, 1.5 million variants out of 251 millions are missense variant. You can actually, another view is to break down the number of variants per consequences, VEP, low, moderate, and high impact. So yes, the vast majority of them are low. You have a few high impact and some moderated, 1.4 million. So in actually Kids First, we as I mentioned, we uploaded the public data sources for population allele frequencies in populations. So we uploaded the TopMed, which is 449 million. 549 million of variants. We have Nomad version 2. We have 1,000 genomes. We're working on adding more and more actually of this kind of database. And here you can see a nice paragraph, Venn diagram actually. So you can see that among the 251 million variants that we have in Kids First, 127 million actually overlap with TopMed and NoMed. And these variants actually are as an allele frequency below than 0.5%, 5% actually. You can modify this, and then let's see on the total that. And now what it's doing now, it's actually querying almost 1 billion. The TopMed, the NoMad, plus the 250 million variants in Kids First, they try to find the commons one and build this Venn diagram. So it takes a bit of time, but it's so bad actually. So the cluster behind this is a cluster of 10 nodes, and if we want more performance, we can just launch a cluster of 20 nodes. And the only consequences is it would be more pricey, but that's it. But the performance with it. Okay, so the entire, so this is the number of variants that overlap between Kids First and TopMed and NoMad. And you see that the vast majority, a lot of TopMed variants are in TopMed and vice versa. You can see 88 million are both in TopMed and NoMad. So in Kids First, we also calculate allele frequencies within our cohort. So we can produce, I won't do it, produce this kind of a NoMad-like table where you have the variants in a given gene and the consequence and the allele number and allele frequency in a table format. So we do that. Gene set now, so we did import a few gene set, and this is just the beginning. We do have OrphanNet, So Orphanet is actually more than almost 4,000 genes, 4,000 gene set for a total of 4,000 genes. HPO panel also have a set, so we have a 77, almost 8,000 set, each one corresponding to an HPO term. And we have uploaded the gene cancer census, cosmic, the development disorder gene from the cipher. And this is only one set with this number of genes. And our intention is we aim to upload genome England, Amin, Reactome, and many others. And then you can see very simple, the overlap between the development disorder genes and cancer genes. And you see that they're still, they have 221 common genes actually between both sets. One important thing actually is that, so as I said, our platform is based on Spark. And it turns out that ALE also is based on Spark. ALE is a tool developed by, it's a very nice tool actually to perform a QC analysis and other kind of analysis on variant data. And so the nice thing here is that we can load our variant database into ALE format using the Python API. And so that we have all of the sudden all access to the ALE, the tools from ALE for our own data. So that's really good. And here I show some examples of QC analysis. So in order to prioritize variants, you really need to do a very good QC before. And this could be done with ALE actually. And then because it's all provided by this nice tool. So this is just two histogram. One is for the read depth for a given set actually. So this loaded my demo set. This is all the variant in my demo set present in the participant from my demo set. And then you see like this, the read depth and the genome type quality. Now some example of analysis. So here from my demo set, I extract some gene hits. So the criteria is that the read depth has to be higher than 10, and the variants are either pathogenic or likely pathogenic in ClinVar. So because we uploaded ClinVar as well, and then you can show an histogram of the number of participants for a given gene. Okay, so for example, the first genes GATA4 is altered in 57 participants from my cohort, and this gene actually belonged to both kids first and of course kids first, but also the development disorder gene panel. And so you can see, and so forth. And you can see here, for example, in orange, this is a cosmic genes present in 10 participants. Remember, this cohort is actually a birth defect cohort. And so I found 2 cosmic genes actually for this birth defect cohort. This just to show that we can do a lot of things actually, because you can execute any Python program, so you can import any kind of library that is existing out there. So, for example, a Python library that does heat map, and then you can plug this to your own data, to the variant database actually. So this is these are the same genes that is represented above, and we do a app map. So on the x-axis you have the phenotypes. And then on the color is proportional, the darkness is proportional to the number of participants. You can zoom in if you want. And so forth. So you see that there's no limitation of the kind of analysis you can do with this kind of Zeppelin notebook. So let me conclude this presentation by going back here. So here, oops. So what are the next steps? Well, we're gonna release this in a few weeks, but because we want to do before some performance tests and data quality control. And the first release will be available for the Kids First user group and the XO1 investigator only. And then we will start integrating feedback, and then we will make a more broader release for a broader public audience. And then in terms of roadmap, short-term development roadmap, well, as I said, we will need to index these variant data using the Elasticsearch so that we can start building a web interface like to search these database, and not only through the Zooplin notebook. So we will do this in multiple phases with different level of complexities for the web interface. We will start probably with a GA4GH like beacon service. So the really yes or no answer. So you have a variant, you want to know if this variant does exist in Kids First. You type it, and then you get the true false answer. We want a kind of normal light summer interface where we may need to get the added frequencies in the Kids First cohort. And eventually we will integrate this data warehouse within our cohort builder. So this will allow us to make very complex queries with both participant and that will return both participant and variant list actually. And we will add more annotation. Finally, I would like to thank a lot my team and people at CHAP, especially Jeremy Constantin, which is our lead software architect at St. Justin and a senior developer. The developer team working at St. Justin with Adrien, Evans, Francis, our UX specialist, Luca Lemonnier, and at CHAP, Alex Lubnowski, which did a fantastic work on for setting up the Spark cluster with Jeremy, and the leads, Yun Kanzu. Thank you.
Thanks so much, Vincent. That was really phenomenal and a ton of work. And as Valerie switches the presentation to Allison, I really just want to highlight, that our goal is to really continuously improve on the user's capacity to go through the types of analyses that you saw earlier in the presentation. Where a user who's interested in something like a neural crest-derived birth defect could leverage neural crest-derived tumors like neuroblastoma to really intersect across data sets, leveraging really this phenotypic context plus the variant environment that Vincent highlighted. All of this then intersects with the user workspace environment that Allison will talk about briefly now.
Yeah, thanks, Adam and Vincent. And, it's kind of as we're going through this webinar, I think I'm struck by, we try to meet the needs across a very wide variety of folks to really accelerate the discovery. And I think, as we roll out the variant warehouse and associated portal features, intersecting that with what you heard at the beginning of the large scale, workflows, especially in the spaces where there aren't standards. I think, the DRC in terms of alignment, it's a very base level variant calling, we do that to provide it to the community, but there's a very large diversity of what people are trying to study, the different disease types, and really what we're trying to do is provide platforms, workspaces, and especially collaborative spaces where you can have both private workspaces, partnered peer-to-peer workspaces, or potentially even consortium or larger groups. And so Cavatica has been the initial cloud-based workspace that we've been integrated from launch. And so I just want to go over some of the kind of key highlights, emphasizing some of the different pieces of it. And then I think Valerie will talk a little bit later on about some of the interoperability work. I think we are very keen to make Kids First data available, not only in Cavatica, but in Terra, as you saw earlier, and other NIH cloud platforms that are coming online. So I'm just going to walk through some real high-level basics, and then afterwards you'll actually hear from Owen and Sharon Plon's lab at Baylor about some of the work that he's done also on some of the structural variant work. So when we look at Cavatica, we really think of data inputs. So one place that you might come from is the Kids First Data Resource Center, but we've also seen many use cases of people bringing their own data, wanting to bring data from places like TopMed, from the NCI Cloud Resource Data Commons. Then, being able to run workflows on different pieces of this. We hear a lot, I want to do a joint genotype on a custom cohort. I want to recall everything to be able to do my analysis. I want to bring a new tool pipeline. How do we support that? And then interactive features. So you saw some of the notebook work on the variants. In Cavatica, there's also different kind of notebook environments. And it really just depends, is your code coming from Python? Is it R? What kind of user? Who are you working with and collaborating with? And we really want to empower all those different. So as you saw kind of from the portal, from the very beginning, we had this ability to immediately bring your files, bring files from Kids First over into Cavatica fairly seamlessly, and then run different kinds of workflows. So for example, if I was interested in the GBCFs that Vincent showed earlier, because I want to integrate that with other variants that I have in my system, on the cloud, et cetera, you can immediately bring that over into the Cavatica workspace. And so, something that, we emphasize the collaboration, we emphasize the ability to share and have these kind of platforms, but something we, I think, don't always focus on is your projects are private. This is really taking some of the local workflows that you had into the cloud environment. This is your own workspace. You can start it on your own and then add people as you want to collaborate. Or if you really, want to keep your own private workspace, that is completely fine. It's similar to a Dropbox environment or a Google Drive environment. You get to choose. You get to drive in your own project, in your own environment, who gets to see that data. The Kids First data is just one entry point into the kind of workflow that you could bring on Cavatica. So as I said from the beginning, kind of the ability to immediately bring over the Kids First data. It's not copying. There's no extra storage charges for you. You can immediately start using it in the environment. And as we're working with other data sets across NIH, in the back end, a lot of this is doing integrated checking. We've been working with the NIH in a new initiative called the Research Authorization Service to really do this more seamlessly across different NIH data resources. Something else I want to emphasize, because it's been launched a little more recently, is that while Kids First is on Amazon Web Services, Cavatica actually has the capability to bring in data both from Amazon and Google. So you can select a volume, if you have your data in the cloud somewhere, and be able to bring it into your own private or shared Cavatica workspace. And so in there's a data volumes feature that allows you to do that. And then more recently, the compute can also be done on Amazon or Google as well. And something we are working on is how does that kind of work a little more seamlessly. But for now, you can choose one and kind of stay there and then integrate downstream data flows. And so similarly, this is just an example of if I had a project called Kids First Analysis, I was bringing my own data. Here's just some example, 1,000 genomes data, for example, but you could imagine it could be your own buckets, either on Amazon or on Google, and be able to bring them into the environment and have them in one workspace where you could bring a new workflow and run it across there. So as we've kind of progressed in Kids First, we've increasingly hardened some of the initial baseline pipelines. These are all available on the platform. They're also on GitHub. And we have some of the basics of RNA-seq, alignment, joint genotyping, and some of the somatic workflows here. But that's just one place. So if you wanted to run the Kids First pipelines on your own data, you could start there. Or as you'll hear shortly, a lot of times people want to be able to bring their own workflows. And so using Docker and the common workflow language, we've been able to work with a number of users to bring their own workflows to the platform and then to run them and scale them across both their own data, Kids First data, other NIH data sets as well. So as you saw these kind of notebook environments, we've increasingly found, I think, there's a reason why everyone has them. They're quite useful, right? So someone can do an analysis, create some graphs and plots, and it's very collaborative. So you can share that with someone, they can review those plots, provide input. I think there's something wrong with QC here. I don't really want to expect those genes, or wow, that's really interesting. What's going on with this one patient over here? And really starting to iterate on that capability of having both the computational as well as the disease expertise in similar places. Obviously, as Adam was saying, we're always trying to improve on this. Nothing is perfect, but it's an initial start. And so this is kind of just a animated GIF of, a lot of times we get asked by some of the biometricians, can I have terminal environments? What kind of notebooks do I have? And so really within the workspace, you can have more command-like environments like you're seeing here alongside of the more kind of notebooks graph style that you saw Vincent show earlier. And then similarly along that line, there's been a recent launch of RStudio and the ability to actually run Shiny apps. So folks who are in the R community, I think, are very familiar with Shiny apps being a very quick way of getting your data, being able to have an exploratory interface and have other people look and have input on it who aren't necessarily writing R code. So this is brand new and kind of a beta feature that are starting to be tested. So again, we are interested in any and all feedback doing this, and we have some kind of examples of the different R Shiny apps that we're starting to see. And I'll mention, again, the level of different kind of collaboration, how you want to do it. It's really, a cloud platform that's supposed to serve you and your research, and really being able to maintain the control of the different kinds of access, the different kind of permissions. But one of the great things is if you do want to share, it's already there. You don't have to transfer the data. You don't have to work through different kind of security and compliance. The platform meets a lot of those requirements. And then additionally, increasingly, we're very keen on how to interoperate and work with other cloud-based environments here as well. So that kind of wraps up my part, and I'll hand it over to Owen. I do want to thank Seven Bridges Genomics, who've been great partners in developing this, especially Jack Giovanni and Michele Mattioni, as well as many of our bioinformatics team who have been working with users trying to understand, your workflows, your pain points, and how we can make this better. And a lot of that has informed the work that you've seen from Vincent's team as well. So it's definitely a group effort. Thank you.
Okay, Owen, you just have to share your screen and then unmute yourself.
Okay.
Looks
Can everybody see it?
Yeah.
Okay, great. So my name is Owen Hershey. I'm a graduate student at Baylor College of Medicine in Dr. Sharon Plon's lab. And today I'm going to talk about our lab's use of Chromatica through the whole genome sequencing analysis of the basic 3 childhood cancer cohort. So in the Plon lab, we're interested in whether germline mutations are involved in inherited cancers, particularly those occurring in children. And we know that germline mutations in cancer susceptibility genes occur in roughly 8 to 10% of all pediatric cancer cases. We also know that there are well-characterized germline susceptibility genes implicated in specific childhood cancers, such as RB1 in retinoblastoma, NF1 in neurofibroma, and WT1 in Wilms tumor. If we look at Wilms tumor and WT1 as an example, the first susceptibility gene was cloned in 1991. And since then, few other genes have been shown to confer susceptibility to Wilms tumor. However, with the recent advent of whole exome and whole genome sequencing, researchers have been able to identify new genes involved in the susceptibility to Wilms tumor, including 4 published last year. This is all to say that not all cancer predisposition or susceptibility genes have been identified. And this novel implication of genes in pediatric cancer is something we in the Plon lab are interested in. One cohort we use to look at these cancer susceptibility genes is the basic 3 childhood cancer cohort. This originally started out as a proband-only whole exome sequencing study of germline and matched tumor samples of 287 children with solid tumors. And the primary goal was to characterize the diagnostic yield of this sequencing methodology. It was not enriched for any one specific cancer type, and the data we found was multiple pathogenic or likely pathogenic variants in genes that were previously associated with specific cancers, such as WT1 and Wilms tumor. But we also found these variants in genes that had not been previously associated with specific childhood cancers, such as FLCN and chordoma. All of this data was published in 2016 and is currently available through dbGaP. Then, through the Gabriella Miller Kids First Pediatric Research Program, we were able to obtain high-quality germline whole genome sequencing for 120 probands and their parents from this original sequencing study. And this started off with the initial goal of discovery and identified de novo single nucleotide variants and structural variants, and another goal of identifying putative pathogenic variants in known cancer genes that may have been missed in our original study. This cohort consists of duos, singletons, but primarily trios. And today, I'm really going to focus on our identification of these de novo single nucleotide variants and structural variants through the Cavatica platform to highlight some of the capabilities of the platform. To start off, we looked for de novo single nucleotide variants, or those variants that only occur in the child and are absent in the parents. We could have used tools like the Broad's GATK toolkit to perform joint calling and de novo variant filtration, but our lab in the Sequencing Center at Baylor has a preferred caller called Platypus, published alongside this paper here in 2014. However, it was not available on the Cavatica platform. We reached out to the team at Cavatica to see if we could upload this tool. In a matter of weeks, this tool was uploaded and able to run in the same manner you would run it on a local system. And we were able to generate our preferred de novo structural or de novo single nucleotide variants. And while we were able to generate these calls, we were also able to create a user-generated workflow shown here that allowed us to rapidly filter these variants and understand and make sure that we had true high-quality de novo variant calls. Then we wanted to annotate them. So we used Variant Effects Predictor tool, which is also present on the Cavatica platform, alongside the up-to-date reference databases to find the pathogenicity scores, the allele frequency, and any published data on the variants we find. Then we had to manually curate and prioritize these variants. We did so by looking for coding variants and determine if they have a functional effect on the protein, and then we ranked them based on their pathogenicity scores, such as CAD and REVEL, and performed literature and database queries for those that were prioritized and ranked highly to determine if they had a potential tumorigenic effect on the child's cancer. And from all of this and the trio sequencing data, we found anywhere between 60 to 190 genome-wide de novo events per person with a range of 0 to 4 occurring in a coding region. This fits in line with published data, and some of these variants we found we're following up on, including one EP300 variant in a rhabdomyosarcoma patient. Then moving on to our de novo structural variant analysis on Cavatica, we had to create our own pipeline on the platform. So in short, we use 5 different callers mentioned below to increase both sensitivity and specificity of variant identification. And we run all 5 callers on each individual, compiling a list of variants per individual. Then we use an overlap filter specifically on the child to make sure that the variants are present by at least 2 callers. And this allows us to account for some variation in the manner of the calling. This pipeline is currently set up for de novo variants, but can be set up for inherited variants. And then we use a set of healthy controls and another filter that requires the de novo variants to not occur in a healthy population. Thus, what we end up with is de novo structural variants that do not occur in a healthy population and are potentially cancer-causing structural variants. So as you can see, this is not the simplest pipeline, and it requires us to use multiple features on the Cavatica platform. For the calling, we utilize the parallelization of tasks that allows us to run up to 80 tasks at once, whereas on a local system, you may be able to run up to 8 at a time. And these tasks also allow us to customize the parameters in the same way you would on a local system. And there are times where these tasks stall and you have to restart, but the Cavatica team has been very helpful in letting us know when this happens and solving the issue, and it occurs very infrequently. And once we generate the calls, we have to go through our de novo variant calling and filtering steps, and this is all done on the data cruncher feature of Cavatica. It's run using multiple complex scripts that are not currently tools. We found this data cruncher feature to be the most helpful feature of the platform because it truly allows you to customize the environment through uploading tools, applications, and packages through the Anaconda system for those that are familiar with it. And while we could turn these bash scripts into dedicated tools like we did with Platypus, we found it to be very convenient to do it on here, but are working with the team at Cavatica to turn them into tools and eventually run them on other cohorts on the platform and eventually make them publicly available. Then when we want to visualize these variants, we use an imported version of Integrated Genome Viewer, or IGV. We could use the genome browser feature of Chromatica, but we found that to look at pair orientation and insert size, this imported version of IGV has been much quicker to use and is easily controllable through the data cruncher feature. And while our pipeline is not a currently available tool, our lab has utilized the feature of uploading tools and sharing it widely on Chromatica. A former graduate student in our lab, Nanad Oak, created a tool that allowed for the analysis of microRNA sequence variation called Admire, published along this paper shown here. And upon request of a fellow Chromatica user, we were able to upload this tool and make it widely available to anyone used currently. So I would like to thank the team at Kids First for letting me present on how our lab uses Chromatica on a daily basis. And if anyone wants to send any questions in the chat, please go ahead.
Thanks so much.
Thank you, Owen. And Adam, I'm handing it back to you.
Perfect. So I'll only speak for a couple of seconds before turning it back, and really just to frame, what I think, on first glance really highlights a very diverse landscape of activities, resources, tools, and opportunities to engage the analysis of Kids First data sets. And I was listening to a podcast earlier this week in which I was trying to sort of align, the challenges that we often face, where we have very diverse communities of, expert clinicians, biologists, computational researchers, bioinformaticians, and how do we both serve the individual needs of that community, but bring them together on behalf of a common set of goals. And the podcast highlighted, the success of Japan winning the silver in a 4x100 meter relay at the Rio de Janeiro Games, where individually each one of these runners had never even run the 100 meter in under 10 seconds. And if you combine their individual score by simply summing their runs, you can see they're nearly at the bottom of the speed for the team. But the key really for their success at the Olympics was they actually worked over and over and over on the handoff of the baton between each of the runners. And by simply optimizing the handoff, they were able to leverage their individual strengths to actually win the silver, defeating actually, some of the fastest teams in the world. And really that's much of what we're trying to convey today as we move through really this methodology of starting with raw data sets that are available to the entire community, transforming them into a standardized information packet that is queryable, downloadable, or actionable. But where we are today, as we're moving forward through this process, since 2018, is really working on this latter part. And so we began really with just the data ingestion and harmonization efforts in 2018, the launch of the very first portal in 2018, where users could implement a file browsing activity and their first Chromatica workspace environments. Vincent and the team partnered with us to build, the first version of the cohort builder and some initial visualization in PDC Bioportal.
Adam, I'm sorry, are you sharing slides? We can't see your slides. We don't know if you're planning on it.
Oh, yeah, I was supposed to be sharing slides, but I appreciate it. So it's actually just 2 slides that you missed. So I'll do it one more. I'll just start over here. This is what the Japanese racers look like, but really what I was highlighting is, passing the baton and then this workflow here. So for us, as we've moved through the different phases of data to information to knowledge to impact, our goal is always to try and pass the baton and create iterative tools of passing the baton between us and the user community and between the user community itself. And right now we're entering this final phase here, really trying to optimize on that interface between information to knowledge creation, as you saw at the beginning of the talks today with the Mayor-Zita Lab, and ultimately to patient impact through these collaborative interactions and partnerships across NIH platforms and user communities. And that's really going to be the key for our success, is the kind of community-based activities that you saw today. All yours, Valerie. Sorry for the late start of the slides.
No worries. That was probably mostly my fault. I'm just taking it back. And actually, our next presenter will be Danielle Winchester from the NIH Common Fund. But I will share the slides. 1 second.
Hello? Valerie, can you hear me all right? Okay, good.
Yeah.
Second. I'm ready when you are.
I'm ready.
Okay. You want to share the slides or maybe it's just taking a long time to load?
Oh, I'm
Oh,
sorry.
that's
We should
loading.
be sharing.
Yeah, now it's up. Okay, while that's loading, hi everyone. As Valerie said, hello. As Valerie said, my name is Danielle Winchester. I'm the Common Fund's Communication and Evaluation Liaison for the Kids First program. I wanted to share with everyone some impact the program had over the past 5 years. Next slide, Valerie. The program released 10 data sets representing a variety of pediatric conditions. You can learn more about these data sets on the Kids First DRC website, the Kids First Umbrella Bio project page, and the EXO1 abstract page on the Kids First program website. The value of the Kids First dataset will be amplified when researchers use and analyze these data to make discoveries that will ultimately improve prevention, diagnostics, and therapeutic interventions for these conditions. Researchers are using Kids First data to answer new scientific questions, as you've seen on some of the presentations thus far. Also, other scientists are using Kids First data to answer new scientific questions supported by NIH funding mechanisms, such as the Kids First R03. From 2016 to 2019, 13 small awards were funded for analyses of Kids First data. 2 R1s were awarded as well. The Kids First program spurred new collaborations with other NIH initiatives such as COMP2 and INCLUDE. Scientists are using Kids First data to provide insight into neuroblastoma, arthrogryposis multiplex congenita, or facial cleft, which you have heard from Dr. Marizita's team, and congenital diaphragmatic hernia. Over 2017 to 2019, a total of 13 papers were published from the Kids First program. RCR, which stands for Relative Citation Ratio, is a citation-based measure of scientific influence of a publication. The average RCR score for Kids First publications, which is highlighted in the green box, equals 1.32. This indicates that on average, Kids First papers have the same influence as an average NIH-funded paper in its field. The findings from the papers outlined in red were presented during a Kids First webinar. All manuscripts that include the use of Kids First data must acknowledge the data sets or sets by listing the dbGaP as session numbers. The acknowledgement statement for Kids First data can be found at the bottom of the dbGaP study page in the data use certification. I provided a screenshot of the statement at the bottom of this slide. We also covered how to acknowledge Kids First data in the frequently asked questions for EXO1 cohorts selected for sequencing question number 5, which is available on the Kids First website. A complete list of Kids First publications can be found on the Kids First website under the Publications Search page, which I've highlighted in yellow. In my next slide, here's a view of the Publication Search page. Next slide. The last thing I would like to share is you can watch past presentations from Kids First investigators on the Kids First DRC YouTube channel. The DRC posted presentations from Dr. Chung and Diskin. And now I can turn it back over to Valerie.
Thank you so much, Danielle. So now I'll provide an update on our strategic planning. So in late 2018 and early 2019, we probed the community, our researchers, our internal NIH working group, and our external program consultants for feedback to help guide the future of Kids First. In this exercise, 7 key recommendation themes emerged, and while we're not ready to announce new initiatives for the last few years of the Kids First program, we're going to tell you a little bit about how we're continually finding ways to address this feedback. So one theme that emerged was the challenge and opportunity for identifying structural variants that may underlie childhood cancers and structural birth defects. Structural variants include large genomic aberrations, such as DNA sequences that are inserted, inverted, deleted, or duplicated within genomes, and you heard a little bit about these earlier today. But sometimes they're difficult to detect with standard short read sequencing technologies. So one way to address this would be to implement long read sequencing technologies for some of these cohorts. This year, we are offering long read sequencing for XO1 cohort projects as part of a pilot. Kids First sequencing centers offer a variety of long read technologies, such as those from Pacific Biosciences and Oxford Nanopore. And the goal of this pilot is to leverage these approaches to uncover genetic variation that may have been missed with short read sequencing or to further inform findings from previous sequencing. So this is an expansion of the cohort sequencing initiative. Another challenge that has become increasingly apparent is the need and opportunity for additional clinical and phenotypic data efforts, such as data extraction, curation, harmonization, and integration to make Kids First data sets stronger and to facilitate the ability to identify similarities and differences across cohorts. The Data Resource Center and partners from the Monarch Initiative recently hosted a webinar to demonstrate the importance of deep phenotyping and to brainstorm ways to improve phenotype data collection, harmonization, and genotype-phenotype analyses. The webinar featured presentations from Kids First investigators who shared details on their phenotyping efforts. Overall, the presentations highlighted the richness and complexities of Kids First phenotypic data, as well as opportunities for integrating diverse data and tools with the Kids First data resource to further empower cross-disease, cross-data type, and even cross-species discovery. As you saw today, phenotype harmonization and the new ontology browser in the portal enable us to view and study the intersection of phenotypes across various data sets. So the more phenotyping efforts we pursue, the more useful the data resource will be to empower new research questions. Given the momentum around this effort, the Data Resource Center intends to host a part 2 of the phenotype webinar later this summer. Another important recommendation that is really core to the goals of Kids First is to enable researchers to cross-analyze Kids First data with other pediatric data. The Kids First Data Resource Center is part of a new NIH cloud-based platforms interoperability effort, which is a collaboration with NHGRI Anvil, NHLBI Biodata Catalyst, and the NCI Cancer Research Data Commons. The goal is to enable and promote end-user analyses of human genomic and phenotypic data sets across these platforms through federation and interoperability. As you've seen during previous Kids First data resource demonstrations, the data resource already has some interoperability with NCI's pediatric cancer from the Target program. And this new interoperability effort is being driven by additional key use cases that our research community, you all, have identified. For example, the desire to cross-analyze data from the Pediatric Cardiac Genomics Consortium that has been sequenced by both TopMed and Kids First. Like the Kids First data resource, each of these platforms are made up of portals for finding and aggregating data, cloud-based analysis workspaces like Cavatica, collections of analytical workflows and tools, and cloud-based data storage repositories. And the goal is to establish guidelines and technical standards that will empower end-user analyses across these platforms. Over the last 6 months, the group has established working groups to address various layers of interoperability, including addressing operational barriers to trans-platform data sharing, testing technical standards for exchanging data across platforms, coordinating training resources for transitioning researchers to the cloud, and piloting FHIR standards for modeling and exchanging clinical and phenotypic data. The groups are making tremendous progress on these critical first steps to achieving interoperability, but additional activities will of course be needed. Currently, all of these platforms use different methods of logging in and managing approvals to controlled access data, and this is one of the major roadblocks to interoperability. The NIH CIT is therefore working to create one unified research auth service, or RAS, that will streamline access to multiple data resources. You saw today how many systems you have to log into just within the Kids First environment to integrate and access data across various sources. So REST will even simplify Kids First data access procedures too. This group is making good progress, and they plan to deploy components of the service for systems that enable single sign-on and modernize communication of DB gap approval information this summer. As we've discussed during previous webinars, we are continuing our collaborations with the Include project and the Knockout Mouse phenotyping project, as well as beginning to engage in emerging opportunities with the Common Fund data ecosystem. And through these collaborations, we hope to continue addressing the recommendations and the needs of the research community. Now, I know we've been answering questions in the Q&A throughout the webinar, so I'm going to go ahead and jump to answering some of the questions we haven't answered yet, but were very common in the registration system. So the first question is what funding opportunities are available. We have an FAQ on our website that summarizes this entirely. But as of right now, the only funding opportunity that we plan to reissue that uses Kids First funds would be a reissue of the Kids First cohort sequencing opportunity. Using funds from various ICs, you can apply to the Kids First R03 PAR for data analysis and data tool development, as well as the Parent R01, and there's also a Parent R03. To validate variants with model systems, we've listed some FOAs that might be relevant to you. And although we're not accepting proposals for our collaboration with Comp2 right now, you can still contact this email address to pursue additional collaborations. How can I get involved is also a common question we received. You're in, welcome to Kids First. Please go ahead and log into the portal and search across the data and play with some of the tools that are available in the portal to everyone. You can also contact the NIH mailbox or the Data Resource Center mailbox for feedback, and you can visit these websites to learn more. Another common question is, how do I access data? Like we've talked about, anyone can log into the portal to search across data, looking at de-identified metadata. But if you are going to want to access individual level sequence data, you will have to submit a dbGaP data access request in line with the NIH genomic data sharing policy. Once you have access, you can check out these resources for pushing the data you have approved access to in Chematica. And during previous webinars, we've talked more about dbGaP data access requests, and so you can watch that previous clip or visit this link as well. And we received a question about how sequence data are released through the data resource. David, do you want to answer this one?
Question that a couple of our users asked was, how are sequences released? And so we collect data from two different groups. So our XO1 research groups provide patient phenotypic data, and as well as we receive the sequencing data from our sequencing centers. And so we at the Kids First DRC, we do what's called harmonize that data, and so get all those different lines of code to line up perfectly with each other, kind of double check everything, and then we release that to the broader community. And so for each sample, we release the complete genome sequence, and that's in CRAM format, as well as we do some initial steps of analysis. We release the unique differences in both VCF and GVCF formats.
We also received several questions about how community researchers can start connecting with each other. Adam, did you want to talk about this?
Sure. As I highlighted, briefly, we're really in that phase where, as you saw in all the presentation, really need to bring people together, not just within laboratories, but across the community. We've made early efforts on this within the portal. So for example, there's a new feature called the Kids First membership, where you can choose to make your portfolio public so that people can find you, connect with you around your interests and activities. Also, you can always share and publicize any of your activities via a variety of different environments. And this is true in almost every single one of our platforms. I saw a question in the chat asking about public and private projects. Yes, you can make your project public as long as they are meant to be public, and also engage in public projects that are already in Chematica. But likewise manage and bring in members of the community into your own membership space, as long as they have the correct controlled access requirements that Chematica will check.
Thank you, Adam. We have just 1 minute left, and I think there's just a final question about, is there source code for the data portal available?
So I can answer that. Yes, everything is public, available, open source. You can find the code on the GitHub repository, the Kids First repository.
Yeah, we can make sure we share that link with you, Andrew. Well, we're pretty much at 5 o'clock, but I will wait until 5 o'clock to see if any more questions come in. Okay, it is 5 o'clock. I do apologize that we didn't have as much time for questions as we wanted, but I think we were able to answer questions in real time using the Q&A. We received a few really interesting questions that we plan to follow up with some of you individually by email. And all of this will be recorded and shared eventually. So please be on the lookout and stay in touch and sign up for the listserv. Thank you all so much.
Thanks, everyone. And thanks to all the presenters.